discussion, which is controversial, not only between the two. So the next speaker would be Alan Hunter-Shane from University of California, San Francisco. And when we cannot ask the question about a clinical outcome, for example, endoresection, no endoresection, because it would take 17 years with the incidence of melanoma in Europe to answer this question, we need to look into different methods. And we went for geneticists and asked the question, what really happens when a melanocyte becomes a metastasis? So please, Hunter, enlighten us.
Thank you, Jens. And thank you to the organizers for the invitation to speak today. I have no disclosures, except for the fact, as Jens pointed out, that I am a geneticist. And I say that recognizing that today's audience is primarily ophthalmologists. But I think that today's topic will be of interest to all of us, and that is the genetic evolution of metastatic uveal melanoma. Allow me to begin by providing some background. 50% of patients with uveal melanoma will eventually develop metastatic disease, and unfortunately, there are no effective treatment options for these patients. Moreover, uveal melanoma metastases can develop months or even years after successful eradication of the primary tumor. Metastases have a peculiar anatomic distribution, as they nearly always occur in the liver, and until recently, very little was known regarding the genomic landscapes of uveal melanoma metastases, and this is because the vast majority of studies were focused on primary tumors. But from those studies, we learn that primary tumors frequently have mutations that activate the GLFQ signaling cascade, gain of chromosomal arm 8q, possibly targeting the MYC oncogene, and either loss of BAP1, commonly referred to as monosomy 3, or change of function mutations affecting SF3B1 or EIF1AX. And so in order to better understand the genetic evolution of metastatic uveal melanoma, we sequenced patient-matched primary and metastatic tumors from 35 patients. Most of these samples are derived from archival material, that is, enucleated eyes and liver core biopsies, from the Copenhagen University Hospital. We next performed next-generation sequencing of 538 commonly mutated cancer genes, and when feasible, we microdissected and separately sequenced multiple regions of the primary tumor. So as you can see in the example case below, on the left is an enucleated eye, from which we microdissected and sequenced two portions of the primary tumor. Melanoma 1 corresponding to the less pigmented area, and melanoma 2 to the more pigmented area. We also microdissected and sequenced the optic nerve as a source of normal or reference tissue. And then finally, from this patient, we microdissected and sequenced his liver core biopsy as a source of metastatic tissue. We were able to deduce the relative order in which mutations occurred based on their distribution across these tissues. The idea being that mutations which are shared across tissues, these likely precede those mutations that are unshared or private to a neoplastic area. And from this distribution, we can draw what is called a phylogenetic tree, as shown here on the right. And so to begin, we wanted to ask whether there were any pathogenic or notable mutations on this tree, and indeed there were. We first found a gain-of-function mutation in the GNA11 oncogene, biallelic loss-of-function mutations affecting the BAP1 tumor suppressor, and gain of chromosomal arm 8q. Specifically, there were 5 absolute copies. Each one of these somatic alterations was present in all of the neoplastic areas, placing these hits on the trunk of the phylogenetic tree. But this uveal melanoma was not done evolving. We further noted higher level gains of chromosomal arm 8q in the melanoma 1 region. Specifically, there were 9 absolute copies. And then we found even higher level gains of chromosomal arm 8q in the metastasis. Specifically, there were 11 copies. So in aggregate, this suggests that stepwise amplification of chromosomal arm 8q occurred during the evolution of this uveal melanoma. To round this case out, we also found a deletion of chromosomal arm 9p in each one of the neoplastic areas, placing this hit on the trunk of the phylogenetic tree. Now, ordinarily, this event would not be notable, but it became notable in this case, and that's because in melanoma 1 and in the metastasis, there was a superimposed focal and homozygous deletion of the CDK into a tumor suppressor, placing this event on the branch of the phylogenetic tree. Now, many in the audience might not be familiar with CDKN2A, and that's because this is a gene that had not been previously implicated in uveal melanoma pathogenesis, but I'll comment this is a very well-established tumor suppressor gene across other cancers. Now, because this had not been previously implicated in uveal melanoma, we too were surprised to find this in our study, but more broadly, we actually found quite a few novel pathogenic alterations in our study, and those are indicated in the tiling plot shown here. Just to walk you through how to read one of these, each column corresponds to a different patient, so there should be 35 columns corresponding to the 35 patients in our study. If there's a mutation affecting one of the genes here on the left, then that tile will be colored red for a gain-of-function mutation or blue for a loss-of-function mutation. So to begin, we were comforted to note that there were frequent mutations in our cohort predicted to activate the G-alpha-Q signaling cascade, as well as mutations affecting either BAP1, EIF1AX, or SF3B1, and gain of chromosomal arm 8q. But we also found a long tail of additional pathogenic alterations, here termed tertiary driver mutations, not previously implicated in other studies. And it is worth asking, why were we able to detect these, whereas most other studies were not? And as you will remember in the previous slide, the homozygous deletion of CDKN2A that we observed was present in just a portion of the primary tumor, and in that patient's matched metastasis, meaning that we would have missed it had we not performed multi-region sequencing of the primary and or sequencing of the patient matched metastasis. And so we believe our ability to detect these tertiary events was directly tied to the design of our study. Before I move on, I want to comment that many of these tertiary driver mutations are in fact druggable. To be sure, historically the most promising drug targets are those mutations that are acquired early during the evolution of cancers, but because there are so few treatment options for patients with metastatic uveal melanoma, it would be prudent to explore some of these as druggable opportunities. Moving on, we next wanted to ask, in what order do these somatic alterations undergo selection? And I've already explained within a single case how we can deduce the order, but we can apply those principles to all 35 cases by essentially asking, for a given genetic alteration, how often does that alteration occupy the trunks versus the branches on phylogenetic trees? That analysis is shown in the stacked bar plot on top. To begin, we found frequent mutations in the G-alpha-Q signaling cascade. Specifically, there was a mutation in this pathway in all 35 patients' tumors. And in all 35 patients' tumors, this mutation occupied the trunk of the phylogenetic tree, meaning that these somatic alterations undergo early selection during the evolution of uveal melanoma. We next noted frequent mutations resulting in gain of chromosomal arm 8q, as well as mutations affecting either BAP1, SF3B1, or EIF1AX. And again, these mutations tended to occupy the trunks of phylogenetic trees, meaning that they too undergo early selection during the evolution of metastatic uveal melanoma. But you'll notice that there are some exceptions to this pattern. What this means is that, strictly speaking, it is at least possible for metastatic dissemination to precede these critical events. As for the remaining somatic alterations, they tended more often to occupy the branches on phylogenetic trees, meaning that they undergo selection comparatively later during the progression cascade. In addition to asking in what order do these somatic alterations undergo selection, we can also ask whether they have a preference to occur in primaries versus metastases. And towards that goal, we applied a simple statistical test, essentially asking whether these genetic alterations were enriched in primaries or in metastases. As you can see, there was a general enrichment in metastases, because our study size was 35 patients. Many of these did not reach statistical significance, but I predict that they will as larger scale studies are performed. In our study, those that did reach significance included gain of chromosomal arm 1q, high-level gains of chromosomal arm 8q, and loss of chromosomal arm 6q. So I presented a lot of information today, and I wanted to take this opportunity to synthesize it into a model that explains the genetic evolution of metastatic uveal melanoma. If we start at the beginning, we know that uveal melanomas originate from melanocytes in the choroidal tract of the eye. We learned today that one of the earliest somatic alterations to undergo selection are those predicted to activate the G-alpha-Q signaling cascade. Now, very little is known regarding the precursor lesions to uveal melanoma, but this is likely sufficient to result in the formation of a uveal nevus or some sort of other precursor lesion. We learned today that the next alterations to undergo selection include loss of BAP1 and gain of chromosomal ARM8q, and it is at least possible for metastatic dissemination to precede one of these critical events, and therefore we propose that micrometastases begin seeding early, and they likely continue to seed throughout the evolution of uveal melanoma. What follows next is selection for a series of additional copy number alterations. It is around this time that the primary tumor is typically recognized and removed, but as we know, metastases can arise months or even years after eradication of the primary. And we learned today that they tend to harbor additional pathogenic mutations, which likely facilitated in their outgrowth. So what are the clinical implications? First, we delineated the order in which somatic alterations undergo selection, and this, of course, offers candidate biomarkers for prognosis. And second, metastases continue to evolve and tend to be more genetically advanced than primary tumors. And so we believe it would be prudent if tissue were available to sequence it in search of druggable alterations. And with that, I'd like to acknowledge everyone who made this work possible, in particular our collaborators from Copenhagen University Hospital, Jens Kilgård, Mette Bagger, and of course my colleague back at the University of California, San Francisco, Boris Bastian. Thank you.
Thank you, Hunter. Are there any questions from the floor? So let me ask you, regarding to the previous talk, we think that the metastatic event happens very early, but can we really rule out that the surgery of the primary tumor could cause metastatic disease from the study here?
Yeah, it's funny. I was thinking about that as the talk was going on. It seems like dissemination occurs early. That's what our study showed. But if it occurs early, it probably occurs early and often. I can easily envision that the surgery could exacerbate that. I think the question comes, if you look at the total mass or number of cells that have disseminated throughout the lifetime of the tumor, what proportion comes from the surgery versus occurred in the 10 years prior? I'm not really sure. This is kind of punting on the question, but I could certainly see an argument in both cases.
So when we looked at the different tumors, could we find any correlation between time from primary melanoma to the event of metastatic disease?
Yeah, so if you look at the different trajectories of how metastatic uveal melanoma evolved, there was a good prognosis trajectory, and I'm not sure if this is the dog wagging the tail or the tail wagging the dog, but there were trees in which the metastasis had a long branch and the primary had a short branch, and that would be evidence that the primary was removed and many additional genetic alterations must have accumulated for the macrometastasis to develop. So again, I'm not sure if that's a useful prognostic marker or if we kind of rediscovered that these good prognosis tumors were, in hindsight, the ones that must have seeded early and just had 2 of the 5 genetic hits required for metastatic disease, and there was a longer time.